Well, thank you very much for being here for the third Turing lecture. And again, once again, thank you, Amit, Melima, and Ram for the invitation. It has been a pleasure. It's been a pleasure to meet so many interesting people. So I'm very thankful for the invitation. So as you know, we've been following this strange path. Instead of starting from neurons and small circuits, we started with behavior. We started with behavior in the wild. Then we went to the biomechanics, and we're trying to use that as a window of opportunity to understand some problems that are interesting from the neuroscience point of view. And in particular, I'm going to be focusing on the problem of the dreams and how to proceed to a quantitative study and analysis of the dream. I think that we're very close of being able to interact with our own dreams, and I would like to show this as an example of a way to do that. Very briefly, for those of you that haven't been in the second lecture, the peripheral system I'm talking about is about how a bird is producing a song, which is a process by which we are sending air through a biomechanical device where there are some tissues that can be set in motion and modulate the airflow, generating sound. So you put this labia together and you have these two sound sources that are interacting to create sound. And as we discussed yesterday, the generation of these signals is nonlinear because the nature of these oscillations is nonlinear, and the nature of the bifurcation that is responsible for those oscillations determines many of the acoustic features and timbres of the sound. Then that is filtered by some linear devices, and eventually you get the sound. And we have been discussing that if you do some smart mathematical transformations, the number of parameters that you have to measure in order to generate the sound is very small, particularly the tension of the labia, which you can control by the activation of these huge syringeal muscles, and the pressure, the air sac pressure, and we've discussed in excruciating detail the mechanics of respiration in birds. And we know that we can measure these two parameters while the bird is behaving, while the bird is singing. So while the bird is singing, we can measure the tension, at least the activity of this muscle that translates into the tension of this labia, and we can measure the air sac pressure. So the name of the game today is going to be to use that in order to see if we can study the elusive world of dreams. So what do we know about dreams and birds? There was a seminal work by Dan Margoliouth in the year 2000, where he recorded the neural activity of one of these nuclei we've been mentioning during these talks, that is the RA nucleus. It's part of the song system. So it's a nucleus that is involved in the generation of these gestures. So while the bird was singing, he could measure with high impedance one neuron of the RA system. So the bird is awake, is singing, and he picks one interesting neuron in RA and he sees some pattern. The novelty that he reported in the year 2000 is that when the bird is sleeping, from time to time, spontaneously, close to that very same neuron, you can catch a pattern of activity which is basically identical to the one that was produced when the bird was actually singing. So the bird is completely asleep, it's having a quiet respiration, there is no sound at all, but you see this pattern of activity, and this replay was found very interesting because it was something that was happening in an animal that needs to learn the vocalization. So the question arises whether really these replays are necessary for learning. So it was a paper in Science that had a lot of attention. And of course, there were many instances where you could see this pattern. And if there is a specific pattern that is also associated to production, during sleep, eventually you see the same pattern. But the interesting thing is that also from time to time, you see other patterns okay, that are not exactly the same ones. And what we don't have is a way of translating some arbitrary pattern into sound. So we still don't know the language of neurons. We can associate something that we measure simultaneously with song, like a pattern that is necessary for song, but if we see something else, we don't know how to translate that into sound. What is the bird dreaming when the pattern is not identical to that was used once during birdsong production? And that was the state of the art, basically, with this problem of how to interpret those that are not identical to the ones used during some production. So, as I was telling you before, during all those years, what we were doing simultaneously was trying to unveil the biomechanics of this process. So we were measuring the pressure and the tension of different syringeal masses together with the song, trying to see how these patterns of activity would be responsible for the specific acoustic features of the song. Until by chance, one day, looking at data with my coworker, Franz Goller, we found something that was very exciting to us. We left one of these birds connected during the night. Actually, we usually kept them connected during the night for the 2 or 3 days of an experiment, but we never looked at the data. And when we looked at the data, and why didn't we look at the data? Because the bird was not singing, was breathing very normally, so we didn't expect any kind of activity in the syringeal masses either. But in the syringeal masses, there was activity from time to time. That means that at night, what we found is that when the bird was sleeping, while the bird was having this very normal, quiet respiration, not the patterns that are necessary for song production, the bird is quietly sleeping, from time to time you see in the syringeal masses patterns of activity which eventually are identical to the patterns that are used for birdsong production. So for us, that was beautiful because we could see the replay in syringeal masses. Let me tell you what we did then. We started to quantify what kind of dreams or what kind of replays the bird was having. So let's say that when the bird is singing, you have these pressure patterns, and you see these muscle activity patterns. Let me split those patterns arbitrarily in terms of the pressure patterns that are necessary for generating different syllables. And what you see are these patterns of activity in the syringeal masses. And here I'm drawing them with different colors. So what I'm going to take now is the activity of the muscle activity during the whole night and look for correlations with the different syllables. So here I'm showing the correlation with the yellow pattern, with the green pattern, with the blue pattern. And from time to time, you see these peaks because you identify a green segment and a blue segment. 2 segments that are identical to the segments that are used. Yeah. Margoliouth measures neurons, and we measure the activity in the syringeal masses. So, and why I thought that that was useful? Because when you measure neurons, if you measure something different than what you measure during the day, you cannot interpret it in terms of sound. But in the masses, we have the mathematical models we discussed yesterday. So now I can listen to the sounds that will be produced by those syringeal muscle activities. So the advantage of having it in the masses is that you have the integrated output of the nervous system. That's exactly the difference and the key point in this strategy. So, so if you look at the statistics during the night, this is what the bird is replaying. Here, in the different colors, I'm showing the number of events that you see where the different patterns are being replayed. So it's not true that always you are replaying the whole song. You are replaying pieces of the song mostly. So here at the beginning, the first hour, you have replays of all the syllables. It's like you dream the whole song if you want. But then it's very cute. He dreams this syllable, the blue one for a while, the blue one for a while, then hear the red one for a while. It is like when you practice music and you practice difficult segments and you practice one segment for a while, then another segment for a while. And at the end of the night, you practice them all. You practice the whole song. So here you have different instances. If you look at the data, typically what you see is hours and hours of this. So these replays are this very 100 milliseconds, 200 milliseconds, 500 milliseconds of interesting stuff in the middle of eight hours that you're analyzing. But here you see the replay of one syllable, the replay of almost all the song, one syllable and something that we cannot interpret, a couple of syllables, and so on. If you do the statistics of this, for example, for this bird during this night, you have a lot of practice of one particular syllable, a second syllable, a third syllable, then a couple of syllables that are practiced together and so on, and just a small number of times you practice a large segment of the song. So when I saw that, I said, well, the first thing I want to do, I don't know whether it's useful or not, I want to listen to this. So I took a segment of the syringeal activity muscle, and that was well correlated with what was produced during the day, and I took segments of the song that were associated to the segments, put them together, and listened to a bird's dream for the first time. It was very tempting to do that. So this is the song that this bird was singing. Sorry. Again. So for those of you that don't work with zebra finch will sing exactly this over and over for hours. But when it's dreaming, it dreams something different. So they practice things that they wouldn't do during the day. We have combinations, repetitions that are not stereotyped. So this is the sound of a dream. So here is some little statistics on the typical things that you find. One syllable, this percentage of the time, syllables plus unknown, two or more syllables, and so on. Some incorrect timing, partial syllables, and complete motifs, only 6% of the time. And I found that very interesting because during the day, this guy won't do anything but the stereotype repetition of the song. During the night, on the contrary, practices the whole thing together only 6% of the time. So what can we do with this? Can we manipulate dreams? What if we could actually listen to them? Can we induce neural and muscle activity patterns using this technique? So remember what I showed you actually 2 times already. This experiment that Anna and I did several years ago that consisted on trying to test the validity of a low-dimensional model. So this is a work where the actual question that we had is whether a low-dimensional model was good enough. But this went into the supplementary of that work. But that's what we actually wanted to do. You remember that we were exposing the bird several times to the birdsong song, and we had these huge peaks of activity. If we use the reverse song, we had nothing. Conspecific, we had nothing. And when we use the synthetic sound, we had responses. So this is what we did with neurons. But of course, we know that when you work with neurons, let's say that you are very close to some projecting neuron that's going to burst just in one instance of the song. It's 10 milliseconds that you're going to record at some particular point and nothing else. So I said, well, why don't we do the same experiment, but now using this idea of measuring the muscles instead? So what I'm going to show you is the equivalent of that experiment, but using muscle activity. So here it is the bird singing, and this is the singing pattern of the syrinx. So this is the syringeal activity. And now what I'm showing you here is what happens when you play the song to the sleeping bird while you measure the ceilings. And what you see is basically the pattern that was used when the bird was singing. So the same thing that we noticed in neurons, we could measure it in the syringeal activity. So you could induce this activity by exposing the birds to a copy of its own song. Here I'm showing you the superposition of several repetitions of the experiment. So as you can see, some of the patterns are very easy to be elicited. Sometimes they are not elicited, and some patterns are more difficult to be elicited. But basically, you have the same shape that you have during the day. So if you repeat the experiment, here it is the bird exposed to its own song at night when you measure simultaneously the muscle activity. Here it is what happens when you play the reverse song. So it's the same notes, but in the reverse order. When you have the conspecific, and here it is when you play the synthetic copy. You can elicit the whole sound. So this is done with synthetic sounds, and we can elicit the activity of the muscles. Now, something that reminded me of your work, or what I thought was had in common with what you described before. I didn't include the transparencies here, but one thing that you can do when you expose the bird to a synthetic copy is you can degrade the quality of the stimulus. You can, for example, generate the stimulus with some noise, for example, and with different levels of noise, or changing some part of the model, and you're starting to degrade the response. But when you degrade the response, you don't degrade it completely, you degrade it by segments. So there are segments that are either elicited or not, but they are complete segments. So whatever sounds like a complex pattern like this could be actually thought as summing two segments, and you could degrade them partially. So you could degrade pieces of the segment. It's not that you degrade them continuously, both of them. That's the reason I was asking you whether these complex patterns you were showing could be decomposed into simpler elements. Now, what I thought about this is that since it is true that you can impose to the brain activity of the bird, of the sleeping bird, this pattern of activity, can you overwrite the dream? So you're dreaming about something. Can I overwrite it? So here's an example where we did that. We had a canary sleeping, and it started to have a dream. So when it started to have the dream, what we did is we started to play the song, the birdsong song, with the idea of whether we are going to interrupt this practice and get we don't know what. So what we get is that we start to elicit the muscle activity that was associated to that particular sound. So the bird is dreaming, it's replay, whatever it was, but with the stimulus, we overwrite it and we control the dream of the bird. Now, to us, this opened a very interesting question, because now we had an easy way of analyzing a question that from the pure neuroscience point of view was very difficult, that is, whether these replays and dreams are important for learning birds or not. Why do I say that? If we want to study zebra finches, you have the map, and we are all working with one or two nuclei because we know the exact coordinates. But if I want to work with Kiskadis or any Tyrannidae, no one knows, not even if these nuclei exist at all, much less where to find them. So how can I address the question of whether these birds are dreaming because they are learning or not? And to me, that's a very interesting question, because there are 10,000 species. These are the assigned birds. These are the ones that need to be exposed to a tutor to learn. But there are, I don't know, 29 other orders, okay, where there is no, well, there are 2 other orders where there is learning, which are, colibríes and loros. How do you call it? Hummingbirds and parrots. Okay, but in general, in half of the species, there is no learning. So what if I take a sabocine, which is phylogenetically close to the sign birds, and they don't need to learn? They have genetically programmed all their vocalizations. Do they dream? Do they have variable dreams? Do they dream at all? So to address this question, we work with a beautiful bird that exists in America. This is a Kiskadi, Pitangus sulfuratus, and it's very common in Argentina in particular. And it has a very simple vocalization. It's called Kiskadi, 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 or bicho feo, bicho feo, bicho feo. In every country, this is a word that onomatopoeically tries to capture the sound of these three syllables. But what these birds do is very different than what sign birds do. The frequency modulation is done by depression. That is something that we unveiled several years ago. But they use the masses for something very strange. They use it to, do you know when a teenager spends 2 hours in front of the mirror to try to look kind of rough? Well, they do exactly the same thing. They put a lot of effort into singing the first syllable slightly out of tune. So here, they have the left side and the right side at a slightly different frequency, and that first syllable sounds a little bit rougher than the rest. And they do it with a lot of care because they're using the syringeal muscles, creating the particular frequency difference to lock these two oscillators at the frequency difference, which is, by the way, from the nonlinear point of view, a non-trivial thing. But they do it. So here it is. And this is the best. In the second syllable, they have a touch of being out of tune. Every Kiskadi does the same. Why on earth evolution found that interesting? I have no idea, but this is what they do. So this is a Kiskadi singing, and from time to time, they will have all these patterns of activity where they are trying to get these syllables out of tune. So what happens when the bird is dreaming? Well, when the bird is sleeping, well, they do that. They have these patterns of activity with a slight difference in frequency, slightly lower frequency. So with 180 hertz, 170 hertz maybe when they are awake, and around 150 hertz when they are sleeping, okay? And the duration is also slightly different. They are slightly larger when they are asleep, so it's like lower frequency and longer. I love that. Here, this is the typical EMG with the 140 hertz, where they create this modulation that gives this rougher timbre to the first syllable. Okay, and this is the association between this modulation and the EMG frequency. I'm not going into the details of how they control these two sides to do that. But you can model this and reproduce the right timbre. So now that we have the model and we have the data, we can synthesize what the Kiskadi is dreaming. And here we have. No, this is a bird I didn't show. So this is a bird that, this is a sabocine. They have a complete different physical mechanism because in oscine birds, when they use the syringeal muscles, they use it to stretch these cartilages, and they stretch the labia that are in between them, changing the restitution constant and therefore increasing the frequency of the vocalization. And in here, the frequency of the vocalization is basically the earth sac pressure. What they do with the syringeal muscle is control the frequency difference between the two sides. So this is a unique.
Equation of the previous slide.
And the equation of the previous slide corresponds to each of these two sound sources, okay? And K, the parameter, is where you will input the EMG data. So in order to generate the sound, you study the dynamics of these two sound sources, you input in here, and what we find is that, first of all, that we can reproduce with the singing EMG the right timbre. This is how the first syllable sounds. And this is the dreamt. So that was super interesting because at least they would practice, but interesting enough, they wouldn't do crazy stuff. They would practice only this pattern of activity. But the interesting thing is that these birds do some other thing. When they are very upset, okay, I don't know if you heard that, there is a very, very fast trill, and they take the feathers out. So when the bird is very unhappy, either because it's upset, because there is an invasion in the territory, or it's very excited, they will generate a different vocalization. It's a trill and a very spectacular display. So one day I go to the lab, and Juan Doppler, who was the grad student working on this, told me, I found the EMG activity of the trill. So, oh, that's beautiful because, it's a second vocalization and it's practicing. Did you do the statistics or something? No, I found something better. So he showed me the movie that we used to check that the bird is actually sleeping with the eyes closed and so on. And this is what he found. When the bird was having these practices, also he would have this huge display. No? So the guy was actually living a nightmare. Here we have it all together. So I tease one and I tell him that he recorded the first nightmare in history. So here you have the pattern of activity, and at the same time, this physical display. So it's having the whole package all together, vocalization and the visual displays of the feathers. This is using the same model, synthesizing the trill. This is the EMG activity. It is used to generate really out of tune syllables. So in order to have this intimidating sound, they also want to sound rough. And they have this first syllable like completely out of tune. So of course, now the program is to try to do this with oscine birds. That means measure these patterns of activity, including them in the model of sound production, and synthesizing the song if you use what is using during the day, or the dreams if what you are measuring is the EMG during the night. Can we do that? Well, actually it's going well. Here is the synthetic sound that we get when we put into the models the measured gestures. This is during the day. So we have K, which is the restitution that comes out of the EMG. And here we have, in black is the pressure. Okay, in black is the pressure. In K is the action of the modulating the frequency, and here the action of the gating. The action of the gating is related to VS, as we were discussing yesterday, right? And here it is what you get with the actual song and the synthesis. So the model is working kind of okay. So now what we're doing is synthesizing these dreams. The next step, trying to see what the bird does if it's exposed to the dreams. So kind of questions that we are addressing now are the following. Well, this is, well, in its way, extending the synthesis to oscine birds, and now expose a sleeping bird to the synthesis of a dream. Because one interesting thing is, why do we do the practice silently? What would happen if you would hear what you are dreaming? So we want to check that. What happens with the learning bird when it's listening to its dreams? And also trigger a dream, because apparently for an oscine bird, dreaming crazy dreams is interesting. What happens if instead of crazy dreams, you would dream stereotype dreams? So are crazy dreams necessary for learning or not? So overwriting dreams will be another thing that we want to explore. So conclusions. To us, the periphery was interesting, as we discussed yesterday, because I think that in neuroscience, it is very important to study the nervous system together with the periphery. When I showed you yesterday that when you breathe, the rhythm of your breathing is the rate at which you have the resonance of the periphery, means that you cannot impose with your brain something that your body doesn't want to do. We have to study the whole thing together. But that's what I think. But besides that point, also that if you want to understand a timbre, a behavior, it is not only necessary to understand which is the pattern of activity that you are creating. You want to see how that would affect the periphery with which you create the behavior. Okay, because some of the features exist because of the timing that is determined by the brain, but some of the features are the result of the brain driving some particular nonlinear model, like this one. The roughness of the solution comes because of the way in which the periphery responds to an instruction that is just creating a frequency. So you have to study the whole thing together, I think. But also, in this case, I want to use the periphery as a window to the central nervous system. I will declare boldly that, of course, since I don't have the money to do optogenetics and buy very expensive electrodes, I will have to make it with very simple electrodes that I can buy. But what I can get with this is the total output of the nervous system, which I think that in some cases is extraordinarily useful. So the periphery can be an interesting window to understanding some important aspect of the central nervous system. So with this model, I think we have an operational way to read dreams. And I think that it's interesting to have an output of what the bird is dreaming. I think it's interesting. So collecting questions, what I'm doing is, well, non-learning birds, do they practice outside the stereotypy? The answer is no. Non-learning birds don't do crazy stuff. Learning birds do. What if we induce stereotype dreams? We have to wait for another talk because I still don't know. What if we let the birds listen to what they dream? I also don't know, but I hope to achieve that before retiring. So I don't have a lot of time, unfortunately, but I have great coworkers who might want to follow this. So let me thank all these coworkers that have been working in these lines of research that I've been describing in these three lectures. Of course, Ana Amador, who is a real experimentalist in the lab, and without whom I couldn't be measuring anything. Franz Goller, who has been teaching us what we do in the lab in terms of muscle recordings. We did help a little bit with his electronics, but he's a wonderful surgeon who taught us a lot. And then Margolias, who has been a big support of our lab for many years. The work with Keith Cadiz and staff was done by Juan Doppler. Facundo Feinstein is the graduate student who has been unveiling the biomechanics of respiration. Alan Bush did the experiments on measuring induced dreams in zebra finches, and Melina Atencio helped us with the biology of Keith Cadiz, sexing them, and so on, because it's a species no one knows anything about, and she's a wonderful and generous biologist. Roberto Vistel is an engineer working in our lab doing the electronics, and is the guy who had been measuring the field results that I described in the first lecture. And these other co-workers are younger and are joining the lab at this moment. And with that, I thank you for your attention. So great question. This is something that we're measuring now. We're pretty convinced they are in REM, because they do have REM, but we have to run a series of experiments we're working on now. So.
Also, so, you know, if you look at dogs, they seem to have lots of movement. I mean, they're moving their limbs, eye movement, all of that happening at specific stages during when they're napping.
Right.
But I can't sort of see what the learning component of this is, especially if it's a very stereotyped song, right? They would have learned it at a certain stage, and beyond that, there is no change. So I
Right.
wonder whether this is a learning or
Yeah,
something.
well, that's an excellent question. I have no idea whether this is important for learning. No idea at all. In general, the people have been promoting this field like it is an important and crucial aspect of learning. I'm not convinced at all. First of all, because as what you say, it seems to be that in order to have a maintenance, you have everything you need during the day. So I don't think that's the reason. Whether you need some practice and you need that practice continuously, that might be an important point, but I don't know. So that is one of the reasons I want to make a bird interact with the sound of its dreams, to see whether that affects the learning or not, or deteriorates the song. My guess is that it won't, because I also think I have serious doubts that it has to do with learning. I do find it interesting in terms of the difference of brains of an ocine bird and a subocine, that subocine have nothing interesting beyond what is used for those two syllables. While in the zebra finch, there are many gestures that are important departures from what they do during the day. So, and that's all I can say.
Is it possible, do birds, in all your experiments that I understand you've done, the birds are practicing their own songs and they've only heard, you play back their own songs to them or synthetic. What happens if you play the song of another species,
Right.
maybe an antagonistic species?
Yeah, exactly. What happens is absolutely nothing. For example, I didn't explain this in detail. This is a conspecific. So this is someone of the same species, but a different bird with a different song. And if you play it, we have nothing. So if you play something of a different species, you get absolutely no response.
Not even if it's dangerous.
No. Not even what?
No, if it's a predator species, something which...
No. Again, it could be the same species, but with a different song, and they won't react at all. So
the bottom panels are not the neuronal activity.
exactly.
Those are the actual activities in the
muscles.
in the muscles.
Right.
So I mean, I would imagine that that if you play the songs of some species that threatens them, nothing would come. Something will happen here, not in the muscles.
Yeah. Well, those kind of things could be interesting if you have, for example, a behavior where some threat would elicit a response, like an alarm call. In that probably would. That's an interesting thing. I never thought of it, but it would be something interesting to check. That could happen, I guess.
So, like, if you take the same individual's part of the song, not jumble, but how much of a segment is required for it to recognize itself?
Beautiful question. So in this experiment that I was describing before, what we did was first to degrade the song gradually and see how it would affect. And what we saw is that this pattern would fall into pieces. Like, for example, you would have this part perfectly, but not this one, or you would have this part perfectly and nothing here. And if you cut a syllable, a complete syllable, it depends on which syllable. Typically, you need to integrate at least for 2 syllables in order to recover this. So, for example, if you play the last 3 syllables, then at the very end, you will have a response. So you have to integrate. But I could never figure out if there is a specific time window, something. So I never studied systematically. So what I reported is the effect of eliminating different pieces of that and how it affects the response. And what I can tell you is that there is an integration, and I didn't quantify that further.
I had the same question. I found it interesting that the spontaneous reactivation could be individual syllables, but if you presented the same syllables in the different temporal order, the reverse song, that you did not elicit that reactivation. So there was some context.
Right. So yeah. So we didn't do a systematic mixing of syllables. What we did is from the song we eliminated A, B, A, B, C, and kept whatever we had in the right order. And what we found is that there was a need of integrating something. And after that integration, you start to have a response. So I didn't play with more sophisticated combinations of that. So I don't know.
So on your first day, you said that the juveniles first learn by listening to some adult, and after in the next year, they will develop their own song, which may be slightly different from the adult they listen to.
Right.
Have you found any correlations between the syllables they repeat and the syllables from the adult song, which are retained next year?
I didn't study that. So I didn't study that. And I didn't do these experiments in juveniles. So although a juvenile can have the same size as an adult, they have more delicate muscles, and it would be right to work with those. And if you try to do any kind of intervention in a juvenile, you disrupt completely its natural learning period. So I could never succeed in doing these experiments in juveniles when they are learning, which would be extremely exciting to do. So, yeah.
So you showed these patterns of the neuronal activity, and these are muscle.
Right.
Are there simultaneous measurements that?
So we don't have simultaneous systematic analysis of that. That is something that Anna and I are working on in the same project where we try to measure whether they are singing in the REM state. We plan to do some low impedance exploration of the patterns of activity in simultaneous, but
Actually, sorry. Maybe from yesterday's lecture, I forgot what you showed completely. Not in the dreaming part and so on, but when they are actually singing, they are alive, during the day they are singing, during that time, are there simultaneous measurements, or even during that time, there are no simultaneous measurements?
no, there are simultaneous measurements
During
when the birds are singing. There are,
And I forgot, did you show those data? Or
no, I
don't
didn't show those
remember
data. Those data were taken by Kyler, no? I think that in University of Chicago, so we did the part of we collaborated with the part of muscle activity, and he was recording HVC and all that data. Yeah, and we have simultaneous data, which we still didn't write up. Yeah. Yeah.
Because that will be, I mean, that will be exciting to see exactly what.
Yeah, of
which
course.
parts match during the.
Of course,
Yeah.
of course, of course, of course.
Other questions? None? Okay, great. Thanks a lot, Gabo, for absolutely wonderful 3 lectures. Extremely enjoyable and extremely informative. Thank you so much.
Thank you. Thank you for your attention.
Minima, you have